Hi, I'm Aakash from Tel Aviv University, and I'm going to talk about an integrated electronic interface for bioelectrochemical plant-based sensors. This work was a joint project by Tel Aviv University and Cartasense Limited. So when we are talking about sensing in plants, what we are usually looking for is information regarding the plant's well-being and health. Does it have enough water and nutrients? Is it under some kind of stress? We can gather this information by gathering data about the plant's surroundings, or we can gather it from the plant itself. The most common type of sensing which we use in agriculture is indirect sensing. This uses environmental parameters to kind of guess what's happening with the plant. We put a sensor, which looks something like this, into the soil, and we sense what's going on in the soil. If the soil is dry, it means that the plant probably needs water. If there are holes in the leaves, the plant probably has some bugs. This is not great. For example, if the roots are damaged, the plants can't take up water regardless of how much is available in the soil. It's also possible that the soil has water, but it's not enough. If we wait to see visible changes, for example, if we wait for some droopy or dry leaves, it's usually quite late to do anything. We want to know if the plant is under stress much before that. So a better option is to do some sensing based on the plant itself. We can look at the thickness and capacitance of the leaves, for example, to see how much water is in the leaves. We can do some spectral analysis to see if the tissues are dehydrated. Dead and live cells have different absorbance and reflectance, so we can tell how much of the tissue is healthy and how much of it is dry. These options can give us good indications of the plant's health and well-being, and they can be used in situ, but most of them are very expensive and they're limited to certain kinds of stress. So, for example, I can use these options to check the hydration status of the plant, but I can't use it to see if the plant is in heat shock stress. So, an even better option is to sense the plant's response to stress directly. It's like asking the plant if it is thirsty, only that instead of words, it replies by expressing a biomarker enzyme. Because this response is a much more basic genotypic response, it's very reliable. We can sense this response using electrochemical sensing, which is very cheap. We can also do optical sensing with, say, fluorescent proteins, but that can be a little more expensive. So we usually prefer electrochemical sensing. Most importantly, this functional sensing can be tailored to work with any kind of stress. It depends on which promoter genes you're working with, but in theory, it can be customized for any kind of stress. We have some previously published work on this kind of functional sensing, which I've mentioned here. So I'll give only a quick overview of the methodology without going into too much of details. So what we do is we use transgenic plants, which express a biomarker enzyme called the GUS enzyme, and they express this enzyme under certain kinds of stress. So what we do is we inject a substrate called paranitrophenol beta-glucuronide. We inject this into the leaf, and the GUS enzyme cleaves this substrate to release an electroactive product called 4-nitrophenol. The 4-nitrophenol then diffuses out from the leaf through the stomata, and it reaches our chip, which is clipped onto the bottom of the leaf, on the bottom side of the leaf. On our chip, we oxidize this 4-nitrophenol, and this oxidation gives us a redox current, which we can measure. So this is what our chip looks like. It's a standard electrochemical system with 3 electrodes: the counter, the working, and the reference. In order to interface with this chip, we need to provide a working voltage between the working and reference electrodes. And because of the oxidation of the 4-nitrophenol, we have some redox current which flows through the counter electrode. This is the real signal that we care about. By measuring the signal, we can back calculate the amount of enzyme that was present in the plant. So in order to do all of this actuation and sensing, we need an electronic system called a potentiostat. So there are quite a few commercially available portable potentiostats. They look something like this blue box. Most of the commercial potentiostats you can buy are around the size of a small loaf of bread. That's too big to connect in the field. They also cost a few $1,000s, which is way too expensive for on-field deployment. They need to be plugged into the wall and use a large amount of power. and you need to have some external system to control the potentiostat. Of course, all of this means that they work really well. They have great accuracy and sampling speed, but that's great when you want to use it in the lab. But for on-field use, it's overkill. So we developed a minimalistic version of this interfacing system, of this potentiostatic system. So this is the first prototype of our system. As you can see, it's pretty small, the size of a credit card. You can mount it on the plant itself. This system is built off using off-the-shelf components. So it's pretty cheap. It costs something like $15. It uses tens of milliwatts of power, which is still quite high, but it's a significant step down from the alternatives. So it means that we can power this minimalistic system using small batteries or solar cells. It's completely standalone, so you don't need a laptop or anything like that. The controller is integrated in the system. Of course, all of this comes at a cost. It has a lower sampling rate and accuracy, but it's good enough for biosensors. So this is the overall structure of the system. It has an analog front-end chip, which interfaces with our 3-electrode sensor. In our prototype, we used Texas Instruments LMP 91000 chip. Then we have an analog to digital converter for quantization of measured data. The single board computer controls the front end, the analog front end, over I2C. It directs the front end to provide the required working voltage and sets the sensitivity of the current detection. So then the front end provides the working voltage to the sensor itself. By modulating what this voltage is, we can execute a number of standard electrochemical techniques. For example, if we provide the voltage step, we can do chronoamperometry. If we give a sawtooth wave, we can do cyclic voltammetry. If you give sinusoids of varying frequencies, we can perform electroimpedance spectroscopy. After providing the working voltage to the sensor, the analog front end measures the current through the third electrode of the sensor. It converts this measured current into an equivalent voltage value using a transimpedance amplifier, which is then fed ahead. In our prototype, we were able to do the sensing at approximately 1800 samples per second. This is slower than what you would get from commercial potentiostats, but it's good enough for most plant biosensors for on-field use. So then the output of the analog front end is sampled by an analog to digital converter. Then the ADC sends this digital signal ahead to the single board computer. Once we have all of the data, we can do some software processing on it. First of all, we need to do some filtering and smoothing to get rid of the noise. We generally use Savitsky-Golay filtering to get rid of high frequency noise without distorting the real signal too much. Then we do some curve fitting so we can extract information about the concentration of the enzyme that we have sensed. The concentration of the enzyme is what we actually care about because that's what represents the amount of stress that our plant is in. Then we can do some more processing steps to make the data more accessible to us. For example, we can plot it to analyze it manually. We can compress it so that we are more efficient in data transfer and storage. To compare the performance of our system, we ran some experiments with a standard test solution with PBS and Ferry-Ferro solutions. And we ran the same experiments with a commercial biologic VSP potentiostat system, and we used the readings from this system as the gold standard. So as you can see here, the green is the measurement from our system, and the red is the commercial system. So as you can see, there is a DC bias, but this can be removed in post-processing, assuming the DC bias is constant. So it's not that big of a deal. But of course, everything is not perfect. Here you can see that the current maxes out at around 9.5 microamps. This is because the 3.3 volt supply voltage limits the output of the analog front end. So we are not able to sense higher currents. This problem can be solved by dynamically adjusting the current sensitivity. This is actually what commercial systems do. So when your current reaches large values, you reduce the resolution in exchange for a larger range. The next issue is how to process and store the data. Of course, we need to store everything in the cloud, and we have two options for the processing. We can either do it on the end nodes, that is on the integrated system, or we can do it later on in the cloud. So we built prototypes of both of these options and tested them. So in this approach, we did the processing on the end nodes and stored all the data on a central server on Amazon S3. In the second approach, we sent only the raw data from the end nodes to the cloud. Then we did all the processing in the cloud with AWS Lambda, and everything was stored in Amazon S3. So the first approach, in which we do processing on the end nodes, is very scalable. It's like everyone does their own dishes. The node which collects the data processes it. So if even if you have a large number of end nodes, it is not a scalability problem. What this means is that the nodes need to be powerful and will use a little more power. On the other hand, if we do processing in the cloud, we can have very light end nodes, but then the processing will take more time and resources. If we consider the scenario that every plant has one sensor and you have deployed it on a large scale in a few hundred hectares of farmland, then you can have hundreds of thousands of data sets coming in every few minutes. Because cloud computing is built by the minute, it can be very expensive very fast. So a good compromise is to do some simple pre-processing on the end nodes and leave the heavy stuff for the cloud. Okay, so to wrap up, functional sensing has a lot of potential in effective sensing of stress in plants. And using this can allow us to make big leaps in the field of agricultural sensing. The problem is that we don't have good interfacing systems. Commercially available potentiostats are too bulky, they're power hungry, and in general, unsuitable for on field use. So, what we did is we designed and prototyped a system which is built entirely using off the shelf components. So, because we are using off the shelf components, it's really cheap, around $15, and it can be manufactured and used very easily. So the system can be used as a better alternative to commercially available potentiostats, portable potentiostats, and it's particularly a better alternative for on-field deployment in a large scale. We would like to thank the Israeli Ministry of Science and Technology, the Israel Science Foundation, the Boris Mintz Institute for Strategic Policy Solutions to Global Challenges, and the Mana Center for Food Security for their support. Thank you for watching.